अब हम बात करेंगे एनिमल्स और ड्रग्स जिनकी हमें ज़रूरत पेश आती है ट्रांसजेनिक माउस प्रेपरेशन में। चार किस्म के एनिमल्स की ज़रूरत पेश आएगी विच आर कैटेगराइज्ड एज नंबर 1 एग डोनर्स, नंबर 2 फोस्टर मदर्स और सूडो प्रेग्नेंट फीमेल्स, ब्रीडिंग मेल्स एंड स्टेराइल मेल्स। Egg donors can be obtained from चार्ल्स रिवर CD1 और इनकी एक स्पेसिफिक एज है। अच्छा, एनिमल्स के हवाले से एक चीज़ मैं यहाँ बताता चलूँ कि एनिमल्स की डिफरेंट स्ट्रेन्स होती हैं, तो वी नीड टू वर्क बिफोर वी प्रोसीड फर्दर कि हमें कौन सी स्ट्रेन के एनिमल्स की ज़रूरत है और हम उनको लेके आगे चल सकते हैं, वेदर दे आर सक्सेसफुल प्रीवियसली रिपोर्टेड और नॉट फॉर द प्रोडक्शन ऑफ दिस ट्रांसजेनिक माउस प्रेपरेशन। तो एग डोनर की एज जो है for should be 3 to 4 weeks old, and once we brought the animals in the lab facility, we need to give them some days so that they can become acclimatized to the given or to the surroundings and they can get out of the stress. So egg donors should be of 3 to 4 weeks age. Foster mother can be from 8 weeks to 6 months old. Then comes the breeding males, again have 8 weeks to 1 year of age. Sterilized or sterile males, which are vasectomized, and for that purpose we need to do the process of vasectomy, and at the age of 4 weeks or 1 month, and then after another 4 weeks we have to see whether the process was complete and successful before we can use them in our practical experiment. जहाँ तक एनिमल मेंटेनेंस का ताल्लुक है, ट्रांसपोर्टेशन और एनी काइंड ऑफ स्ट्रेस कैन कॉज द फिजियोलॉजिकल चेंजेस और कैन ब्रिंग द change in the behavior of the animal. So whenever the animals are moved to a new facility, even from the animal house to the lab, from the source of purchase to the animal house, they must be given some time to get relaxed, to get acclimatized with the laboratory atmosphere, so that we can use them with the experiment. And the food is provided to these animals at libitum, and water is also provided at libitum. एनिमल्स के बाद हमें कुछ ड्रग्स की ज़रूरत पेश आएगी टू परफॉर्म द टू चेंज दिस द फिजियोलॉजिकल कंडीशन ऑफ सम ऑफ द एनिमल्स, लाइक हमें प्रेग्नेंट मेयर्स सीरम की ज़रूरत पेश आएगी सो दैट वी कैन practically introduce some conditions which reflects the pregnancy situation in the animals. Then this serum is diluted in the sterilized balanced salt 50 units per milliliter, and aliquots are stored at minus 20 degrees centigrade. The second hormone which is required is human chorionic gonadotropin, available from Sigma or some other commercial source. It is also diluted at the concentration of 50 units per milliliter in balanced salt solution, and the aliquots are stored at minus 20 degrees centigrade. Then we need Nimbutol, that is used for anesthesia when we are going to perform the surgery, when we are going to perform the dissection of the animals. Then we need this solution. Hyaluronidase is required to disrupt the isolated eggs from the donor mother. So we need it in a concentration. We need the 50 microliter volume in Whiten's medium. Silicone oil is going to be used to reduce the rate of evaporation of the Whiten's medium during the process of microinjection. The oil is autoclaved so as it should be sterilized, is equilibrated with the Whiten's medium. 1 to 7 is the ratio. 1 part is going to be the oil, 7 parts are going to be the Whiten's medium, and this oil is going to be stored in the sterilized sealed container. Then we need gases. So as the eggs or the fertilized egg should not undergo hypoxic conditions. So we need oxygen, carbon dioxide and nitrogen mixture that is passed through the water and is provided in the environmental chamber. Antibiotics like penicillin or streptomycin is required so as the infection or the contamination can be avoided.